Bacteria are probably the most basic forms of life on this planet and are adapted to survival in a wide range of environments. Everywhere there's life, there are bacteria. In fact, in some places, they're the only forms of life present. There are bacteria which feed upon the living and dead tissue of plants and animals, and there are those which feed on inorganic material. The bacteria which transform ammonia to nitrite are a good example of the latter, and the bacteria which cause disease are a good example of the former. Bacterial diseases in fish are fairly common. After ammonia and nitrite toxicity, they account for more deaths than the parasites we discussed in the previous video. The bacteria that can cause disease on fish are usually living in the water and can often be found in the fish's system without any sign of disease. Most bacterial infections are secondary to something else. Something stresses the fish, its immune system is compromised, and the fish can get an infection. Internally, bacteria infect the fish systemically, usually in the blood filtering system. That is, the kidneys, spleen, and liver first. From there, the bacteria can quickly spread to other tissues such as the muscles or the fins. By the time we start seeing bacterial damage in the muscles, the fish's entire system is usually in trouble. Externally, they usually invade tissues which have been damaged by improper handling, such as the fins and skin.
Diagnosing bacterial diseases is easy in some regards, but very difficult in others, unless we get the fish to a laboratory.
If the bacteria is on the outside, we see signs such as fin rot or large open sores. Red, hemorrhaging areas are classic signs of bacteria, but won't tell us which bacteria we're dealing with, or more importantly, what's the best treatment. When infected internally, the fish may just stop eating. Sometimes they will start building up fluids in their body, which will give them a bloated appearance or cause their eyes to bulge. But sometimes the fish may show no signs at all until death. Knowing that we have a bacterial infection is often the easy part, but knowing what bacteria we're having a problem with is difficult to assess.
The problem with identifying bacteria is that they're so small and so similar in shape that you can't identify them even using a microscope.
With all that said, we're going to go through the common bacterial diseases of fish, ways that you can make an educated assumption of which major type you might be dealing with, and what are the best preventions and treatments available.
First off, always check for water quality and parasites before assuming that your problem is a bacterial disease. Check the water quality and make any changes as necessary. Next, look for parasites and treat for them if it's necessary.
Bacterial infections are often caused by the stress that poor water quality or the parasites have been causing. A good water change and getting rid of parasites will often allow the fish's immune system to take care of the situation, even if a secondary bacterial disease is involved. Remember, your fish have evolved in an environment full of opportunistic disease agents and have developed a sophisticated and powerful immune system because of this. When bacteria are involved, parasites can be added as one of the environmental conditions which can cause disease. Bottom line, clean up the environment first. The best and most assured way of diagnosing bacterial disease is to seek the help of a professional. In a laboratory, we culture the bacteria, taking samples from the fish's organs, such as the kidney, liver, spleen, and even the brain. Within a day, we'll have growth of small colonies if the fish is infected. From this sample, we perform two other processes. Samples of these colonies are submitted to a series of tests, and depending upon how they react, we can tell exactly which bacteria we have. At the same time, a separate test is performed to see what antibiotic is best to use in treating it. A bacterial lawn is established on a petri dish with small discs of different antibiotics placed on top. Within a day, it's clear to see the difference between the reaction of the bacteria. If the antibiotic is effective, There will be an area surrounding the disc where the bacteria don't grow. If not, the bacteria will grow right up to the disc. In between these two, we sometimes get partial growth, which indicates that the bacteria is somewhat resistant to that antibiotic. This whole process takes 2 to 3 days to complete, but at the end, we know exactly what bacteria we have, and more importantly, what's the best treatment available.
Well, in the real world, I know that you're not going to run to a veterinarian or a laboratory every time you need to treat for bacterial disease. And with a modern medicine shelf available to the industry, you're not going to be able to resist using an antibiotic. With that said, we'll teach you how to use antibiotics properly, some alternative treatments to antibiotics, and how to make an educated guess on what the best treatment will be.
Bacteria can be broken up into 2 major groups, gram-negatives and gram-positives. This has to do simply with the way they react to a gram stain, a diagnostic test. Most of the bacteria we encounter with fish are gram-negatives, such as Aeromonas, Pseudomonas, Columnaris, and Vibrio. While fish do get gram-positive infections, such as Streptococcus and tuberculosis, they are rare. As we'll see, this is important because different antibiotics are known to work better on one or the other types.
Aeromonas are extremely common bacteria. In fact, they account for most of the disease cases we see in freshwater fish. You will find them on marine species as well. Aeromonas hydrophila and Salmonacida are the 2 most common species we encounter. While they belong to the same genus, they behave and present themselves in much different ways.
Aeromonas hydrophila is truly opportunistic. Living in the water and soil, it only becomes a disease agent when the fish's immune system is compromised. Aeromonas hydrophila infections produce red, hemorrhagic areas in the fins, open sores on the body, fin rot, and a buildup of fluid in the body called ascites, or dropsy. Internally, the gut is often filled with a yellow fluid, the liver is off-color or mottled, and the gallbladder is enlarged. The gills can also be damaged, and you might see rotten-looking areas there. Aeromonas salmonacida can cause all of the above signs, but is more commonly associated with ulcers or sores which appear red and enlarged. Aeromonas salmonacida is considered a pathogen whenever it's found, and is a severe problem in cold water species such as goldfish and trout. Gram-negative bacteria, Aeromonas infections, are usually quite sensitive to a good broad-spectrum antibiotic. Vibrio infections can be viewed as the marine equivalent of the Aeromonas, but are also found in freshwater fish. They cause the same type of lesions and signs as Aeromonas, including fin rot, hemorrhaging, and open sores. Another opportunistic bacteria, Vibrio, is commonly present in the environment and appears only to be pathogenic when the fish is stressed. Vibrios are gram-negatives as well. Therefore, treatment with a broad-spectrum antibiotic will usually take care of the infection. Pseudomonas also presents itself in much the same way as Aeromonas and Vibrio infections. Pseudomonas fluorescens is rather interesting as it will fluoresce under a UV light. Pseudomonas is almost always considered a secondary disease agent because infection only occurs when the fish is stressed or its immune system compromised in some other way. Another gram-negative bacteria, Pseudomonas infections, are also treated with a good broad-spectrum antibiotic. Flexibacter columnaris is another common gram-negative bacteria. It's found primarily on freshwater species and usually shows up as a gray, cotton-like area on the mouth, fins, and skin. Columnaris is unique amongst the bacteria that we find on our fish because it's very large and you can identify it with a microscope. At a magnification of 400 times, columnaris will appear as haystacks of rapidly vibrating, flexible rods. It's easily one of the largest of all the bacteria we find. While columnaris can be identified from a skin scraping or a gill clipping, sometimes the infection is systemic, which makes it difficult to find for the average person. Columnaris is also present in the environment, and some fish may actually be carriers. Most normal, healthy fish are actually quite resistant to columnaris disease. Usually, columnaris gets started on the outside of the fish, especially on the gills, and therefore treating with potassium permanganate has been effective in controlling infections. Another gram-negative, a broad-spectrum antibiotic treatment is the first choice.
There are other gram-negative bacteria such as Flavobacters and Edwardsiella. Edwardsiella is an important disease agent in channel catfish, but it's rare in ornamental fish. Most of the gram-negative bacteria are somewhat to completely opportunistic, so if you keep the stress to a minimum, disease usually will not occur. The gram-positive bacteria as a group are generally more pathogenic in fish, but fortunately are also less common. Most of them are true obligates, needing a fish or some other host to survive. Amongst this group, the two we see the most commonly are Streptococcus and Mycobacteria. Streptococcus is almost always found as a systemic disease. Because of this, it's very hard to diagnose. Infected fish may become lethargic and stop feeding. Eventually, Streptococcus may infect the nervous system and the brain, causing odd behavior such as spinning. External signs can be similar to other systemic bacterial disease, including hemorrhaging and ulcers, but usually these signs are absent. Death rates in populations infected with Streptococcus bacteria can be very high. Added to this, Streptococcus can be extremely chronic in a system or a population of fish, reappearing whenever the fish are stressed in any way. It usually doesn't respond well to any of the commonly used antibiotics. Erythromycin is often used in fish, but presents some problems. It's extremely unpalatable to the fish, so it's difficult to get them to eat it when mixed in the feed, and its absorption by the fish when used in a bath treatment is suspect. If you suspect a strep infection, the best thing to do is to isolate the infected fish and seek the help of a professional if the fish don't respond quickly to your treatment. Mycobacteria infections in fish were formerly called Piscine tuberculosis because of the characteristic tubercle lesions it causes. Mycobacteria infect both marine and freshwater fish. It's a chronic disease which may take years to kill the host, especially if non-vital organs are infected. In contrast, if the infection is in the heart or the liver, death can occur rather quickly. Outward signs include lethargy and failure to eat. Infected fish will often separate from others and slowly waste away. Internally, you can often see the tubercle lesions with the naked eye. There's no economically effective treatment for mycobacteria, and infected fish should be destroyed. Tanks, nets, and other equipment should be thoroughly disinfected with bleach or a strong quaternary ammonia compound such as benzalkonium chloride before any new fish are introduced. While fish appear to be fairly resistant to infection, the fact that there's no effective treatment calls for extreme care when dealing with mycobacteria. We'll discuss in detail in the last video of this series specific antibiotics, their dosages, and how best to administer them. But while we're talking about bacteria, it's as good a time as any to broach the subject of antibiotic usage. Many of the bacterial diseases of fish, in fact, bacterial diseases of all animals, including humans, have become resistant to antibiotic treatments that normally should do the job. The two main reasons for this is overusage and improper usage of antibiotics.
Bacteria are quite good at mutating when their survival is at risk. If a population of bacteria is exposed repeatedly to low dosages or dosages that don't last long enough of a given antibiotic, there's always a chance that some will survive. These survivors can build up a resistance to that antibiotic, so even if the treatment is corrected, they won't respond. The most common mistakes made are: 1, the dosage is too small; 2, as soon as the fish get better, the treatment is stopped; and finally, the problem wasn't bacterial disease to begin with. Never start an antibiotic treatment until you've eliminated water quality and parasites as a possible problem, and never stop a treatment until you've completed it. With what we've just covered, one can begin to see the difficulty with properly treating bacterial disease. Knowing you have a bacterial disease is sometimes the easy part. If the fish have red, hemorrhaging areas, open sores, rotting fins, bloating, or eyes which bulge, you probably have a bacterial disease. The outward signs are sometimes the same, regardless of the specific type of bacteria. So unless you use the assistance of a professional lab, we have to make guesses on which type we have. Then we have to select an antibiotic, knowing that there's a chance that the bacteria will be resistant. This is why more and more workers are starting to use the help of professionals, such as veterinarians and fish pathology labs. It assures you that you know the bacteria is a problem, what bacteria you are dealing with, and what antibiotic is the right one to use. Most of the bacteria are truly opportunistic and only create disease when the fish are damaged or stressed. The first thing to do when you know you have a bacterial disease is find the problem which allowed the bacteria to infect the fish and fix it. Perhaps the greatest tool we have for bacterial disease is prevention. Maintaining good water quality, good nutrition, handling the fish gently, and keeping the parasites off and out of them will almost always keep the bacteria at bay. Quarantine of new or diseased fish will prevent the spread of bacterial disease, and sanitation will reduce the number of bacteria which are challenging the fish's immune system.
As with everything else that we've discussed in this series, the best treatment for bacterial diseases is a good understanding of what makes a fish healthy and keeping them that way. Think about how bacterial diseases get started and stop them before they get ahead of you.